Okay, so now I'm going to stop this video. In the next one, we are going to start talking about chromosomal rearrangement. Yeah, so we already know why we study cancer, then what is cancer. That from we see that there are changes in the cell numbers, in the cell nucleus, in the controlling the cell cycle, and that there are different approaches to study it. Experimental approaches in silico, in vivo, in vitro, and we talk about one of the more important, that it was the in vitro one in the past, that it was the only way that we could make actual experiments. Right now we can use also in vivo, but in the past, like with model organisms, but in vitro was very important, and not only for cancer, and HeLa cells were very important. In the next section, we are going to talk about the chromosomal rearrangement, so focus on the genetics. Okay, so I'm going to stop this video, and we can start and you can play the next one later. Okay, so now we are going to start talking about chromosomal rearrangement, yeah, and so when I know that here you need to remember what is a chromosome, what is a karyotype, you need to remember uh, um, some information that you study in genetic revolution, and some of you already study in genetics or in other courses, but that is very simple. So if you remember this word, yeah, karyos mean nucleus, yeah. Um, so this is why eukaryotes, yeah. So here, a karyotype is an organized profile of a person chromosome. Yeah, so you study what type of things are in the nucleus. Yeah, so you have chromosomes and you divide those chromosomes based on different characteristics, one on different features. Some of them, for example, is the size of the chromosomes and how big or small are each one or the of the arms of a chromosome. And this is very important when you, for example, compare. It's a way to identify a species, is a way to identify diseases. So karyotypes, and in particular, this was a very important tool, that it was the genetics, that it was used all the time. Okay, so remember, chromosomes can be isolated and stained and examined under the microscope. Yeah, so this is why we're able to see them. In one of the first one that we saw was um, the polytenic chromosomes, and we are going to talk that in genetics. If not, please remember that name. So here you can see the karyotype. So here you see the number 1, that is very big, and if you see that they start getting smaller, 21 and 22, and here of course the X and Y at the end, that they are the only ones that they are slightly different. In humans, they determine the sex or influence sex determination. Okay, so here, of course, the chromosomes will be they will not be color coded, just to make it easier. And although there are stainings to, as I said, there are stainings that allow us to see it much easier, just for teaching purposes. Your textbook will show something like that, and also in the video lecture you will see something color coded like that. But the idea is there is 23 pairs of chromosome, and those are. Now, what do you think about? How do you think that they will be? It will be a chromosome of a cancer cell. Yeah, and I want for the assignment, that you have to upload the notes of what you did today. I want that you draw or write what do you expect to see in a chromosome of a cancer cell. What do you expect to see in terms of numbers instead of components? Do you think that there will be chromosomes missing? Do you think that there could be duplications? Do you think that the chromosomes are going to mix, or do you think that which kind of things you expected to see in a cancer cell? Okay, so number, In humans is 23. Appearance, so you divide it by size and position. In terms of chromosomes, normal chromosomes, you have a centromere, you have a short arm, and you have a long arm. Yeah. So how do you think? What do you think that happened in chromosomes of cancer cells? Do the number change? The sizes would change? Do you think the position of the centromere, the arms will change? Okay, so this is a classical example, again color coded, of a karyotype, yeah, of a profile of a chromosome of a cancer cell, yeah, and so there are so many things happen here. So if you see, is there a difference in number? And look, so here you have excellent. One is here, but look what happened with the number 3. You have one here that is very long, and you have 2 here that are very short. Yeah, and look, do you see the color? One went here, and another one went here. Yeah, so there was a translocation. Yeah, it moved in trans. Yeah, I don't want to confuse you with other words, but yes, it moved from one region in and attached to the other one. So there was a chromosomal rearrangement. Yeah, and if you see, you can start seeing that there is a lot of mixes and sizes on chromosomes. There are some that have more than 2 copies, so this is number 2 is not the only is not the exception. Number 3, look number 8, number 12, look number 16. It has so many combination of things, yeah, and there are the I don't know 1, 2, 3, 4, 5 of the same copy. The appearance, so do you see any difference? For sure, I think you see that although number 1 is still is the largest one, is very big. Look what happened with this one is, yeah. And there are other ones that are very small. Yeah, look, this one number 5 should be much bigger, and now it's very tiny. So there is a lot of changes. And super important, there will be interchromosomal rearrangement, and this is what you see as I show you with the color mixing, and there are different types of chromosomal rearrangement. And I remind you, I don't mean that every time that you see a chromosomal rearrangement, there will be something damaged, yeah. Although I'm sure that you know that, for example, trisomy, when you have 3 chromosomes, in humans, they could be a problem, or when you lack of a chromosome, they could be also a problem. However, if you compare, this is just a sign of change. Yeah. So, for example, if you compare chimpanzees and humans, you will see that the genes are very similar, but the chromosomes numbers change, and the chromosomes move a lot. So they are reorganized during evolution a lot. And if you plate, you have different karyotypes during evolution, you will see that although many of the genes still are there, they move from one chromosome into another one, and it's something very common. So a change in a chromosome, in a karyotype, it could lead to diseases, or it could lead to evolution. It's just showing that there are changes. Okay, so I'm sure that some of you still remember this, or some of you will study this in more detail in the genetic course. So we here have different chromosomes, different schematics of a chromosome. And remember, we have stains that generally they show us bands, yeah. So pieces of DNA that stains better. So depending on if you have a lot of A and T's or G and C, depending on the stains, you can create different bands. And humans, using cytogenetics, we became very good at identifying these different bands. So even in the beginning, although we didn't know uh, the DNA content in terms of which gene on how many bases, we can actually see bases, and we can see, for example, here you have a big band and a tiny band. So here you have again a big band, and here a tiny one. I'm going to put a T, and here you have another one, a big, and here you have a tiny one. Yeah, and if you measure the sizes, it looks like they are the same. So there was a duplication, and so one type of chromosomal rearrangement is that there was a piece of the chromosome that duplicates. So you have duplicated different parts of different genes. Yeah. Now we can understand what actually duplicate there. So this is one type of chromosomal rearrangement. I want that you see this one, inversion, and if you see here, it's the name tells you everything. So here you have a big one, yeah, and now the big one is here, yeah. So you move here, and here you have let's put a small one, not a tiny, small one, an S here, and here you have another S. So now it went the S here and the S here, they went up, yeah. So they you find an inversion in the genetic information. Sometimes this doesn't have any change. Yeah, sometimes the duplication can not have any effect. However, if you have a gene that promotes cell proliferation, and now you have two, it could create problems. Or it could be good if there is a mechanism of control. It can also help to control. This chromosomal rearrangement, what it means is they can produce change, or here in those two cases, you still remain with the same information. However, if you see here in the green one, the next one, we have deletion. And deletions, they can be very damaging because you could be missing a part of a chromosome. And if you remember, we saw many types of deletions. Now we move into the next one, that is an insertion, and we also saw pieces of insertion. So here, this piece went to the other chromosome. So you cut and pasted and inserted in another chromosome, and this is different from this one that you find here, that is translocation. When here you have one that is pink, purple, and now you mix it with the yellow one. So here you have pink and yellow, and the other one that it was in the beginning only yellow now has a combination between yellow and pink. Again, this is pretty common during evolution. However, it's a risk to be moving the information. Yeah, you can be breaking genes, or you can move the genes, and now the gene regulation can change. So remember all this information that I show you here, that there are chromosomal rearrangement. That means that pieces of the chromosomes are missing, are duplicated, or there are extra copies that they can move around. And remember that the effect can vary. Yeah, they depend on which chromosome pieces are involved and how they are rearranged. Yeah, so in other words, we need to make experiments and see what kind of things happen. And to see what was the implication, yeah, of sometimes deletions have no effect if the genes are not important, or for this type of cells, yeah, that are not all the genes are expressed in all the cells. And there are many types, and I show you here duplication, deletions, inversion, translocation, and insertion. Okay, so there will be questions of the quiz related to those topics, and in genetics we will do a little bit of math, trying to, what happened when you move. There is a translocation and the size of those translocations, and some information, and talk about more different diseases in terms of with this chromosomal rearrangement. But for cancer, this is what I want that you remember. Okay, now I want that we practice what we just learned. Yeah, so let's try to answer this question. Which of the following is not a structural variation of chromosomes? So we already talked about deletions. So yeah, this is correct. We talk about duplications. Yes, if you remember, is when you have a deletion, is that you are missing. Now you have, for example, a chromosome. This part is missing. Yeah. Duplication. You have a chromosome now that it has maybe double part. Yeah, that it has now this one and have an extra piece. Translocation. Do you remember what is a translocation? That now when you have the pink and the yellow one and they exchange information, yeah. And translation is actually not a translation is not a type of chromosomal rearrangement. If you remember, translation is something different in biology. So when you go from RNA to proteins, yeah. So pretty easy, and there will be more questions like that that they're not complicated, but you will have just to remember what are the different type of chromosomal rearrangement. And here you can see the answer. This is what I already showed you: duplication, inversion, deletion, and none of those is different translocation from translation. Okay, so why we talk about this chromosomal rearrangement? Because you will see that when we start studying cancer, so here now I'm showing you that in 1959, when you watch the lecture, when you watch him talking about this, he will tell you that we start recognizing that some type of cancer, the cells of those type of cancers, could be different. So here you have an example that is this Philadelphia chromosome that it was discovered in that city. This is why it's called Philadelphia, and it's a tinier chromosome that is found in this type of diseases. Okay. So he will tell you more about that. I just wanted to give you the tools to understand much better what is a chromosomal rearrangement and how they appear. And later he will tell you that it's not only important to identify things, but also to understand in more detail what is happening. So the scientists, for example, found that this is a translocation. If you remember, there is exchange between two chromosomes, between the 9 and the 22. Yeah, and it always happened like that. Okay, so very important that we were able to identify from a very general perspective what it was happening. Okay, so now we finish. Now we know what is cancer. We talk about the different chromosomal rearrangements. We know that the information is moving, and this movement, reorganization in the chromosome is from a very general perspective can have no change, or it could have different changes in terms of evolution or in terms of the diseases. And in cancer, is very obvious. And in the other video lecture, you will be able to see the how 50s and 60s thanks to stainings and cytogenetic, we start finding all this rearrangement. In the future, like right now, we are not only focused on chromosomal rearrangement, but actually sequencing each one of those tumors, yeah, and see at the genetic level what is happening, like at from a gene perspective, not only from a very general chromosomal perspective. Now we are going to talk about transformation, that this is related to virology and also to cell biology. So in the video lecture that you will watch, I want that you pay a lot of attention to look this date. We know that cancers can have influences in our body, and some of them don't have any effect. However, other ones that affect specific regions and specific cells can lead to cancer. Yeah, and we know this since a long time ago. So in the video lecture, they will tell you all the information. I'm just going to give you additional information just to remind you of what is a virus and the process of transformation. Okay, then we'll also talk about this gene that is very important. Yeah, this SRC. You will find it very like in many type of cancers, and we will talk about that in the following lectures. Okay, so to remember the process of transformation, here we have, and I'm sure that you know this much better now than me. You have the virus, and here you have the host cell. And what is going to happen is that due to the process of infection, the virus is going to anchor to a receptor of the cell, and they are going to recombine, they are going to mix the information. Yeah. Generally, during the infection, after the infection, the virus leaves the cells, yeah, and the DNA of the virus left already. However, there is a process called integration in which sometimes the virus can attach to the cells and make a mistake, and it remains there. And as a result, if you see here, now you have a combination between the viral DNA and the normal DNA, and this could produce that damaging a normal gene. Yeah, the now a protein is disrupted, so it loses its function. We could also change the regulation of a gene. So now, for example, if you're it's a gene that is controlling cell proliferation, now you can start having a lot of cells that are reproducing without any control. Okay, so again, this is something that it has been established, and there are many viruses that we know that when they interact with normal cells, they generally don't produce any, like they not always produce cancer, but in some cases they can attach, and not in all the cases when a virus integrated to our DNA produce cancer, but in some cases there are some viruses that we know that they do. So this SRC is one of those cases, and the human papillomavirus, the HPV, is another case. Again, I have shared you multiple times, so I just remind you that the way that we study archaeology, yeah, to study archaeology in viruses is through bioinformatics of our DNA. So we go and study if in our genomes there are multiple gene viruses that have got stuck in our integrated in our DNA, and we can go and see if it integrated before of the when we have the common ancestor with the chimpanzees. We can go and see if it has a, or if it integrate before when we share a common ancestor with other primates or with other mammals or with fish. And we have found that there are many viruses that are attached that have integrated into the DNA during several times in the course of evolution. Okay, so this is something that it happened and it will continue to happen, and in some cases, again, it can lead to evolution, or in some cases it can lead to diseases, and but generally it has no effect. So this, remember, infection, recombination. In some cases there is no effect. In our cases there could be integration, and they could be in some cases tumor formation. Okay, let's try to answer this question that is very easy. In which type of organism is the genetic transformation using viruses most commonly used? So I want that you think in plants, animals, fungi, bacteria, or none of those options. So this genetic transformation, when it's used, it can be used in all of those ones, but where is the most common one? And the most common one, and we will study that in genetics, it has different names, but the idea is that it's pretty common that to during biotechnology study the field of biotechnology, and in many industries they use just viruses all the time to insert information into bacteria. Yeah, so it's a common way, a simple tool that it has been used for a long time to design some viruses, virus DNA, and infect cells, and it's very efficient. Okay, so bacteria is a common way. Bacteria is an organism that genetic transformation is used in most commonly. And remember, in genetics we will change a little bit the names, and we call this bacterial transduction. Here in the process of transformation, we are just talking about that the cells transform into something new, yeah, from a cell biology perspective. Later in genetics, we are going to talk in terms of how this information is transferred, and there will be other names. But the idea here, what you need to remember, is that the cell is transforming into something new from a cell biology perspective. And if you have any doubt, let me know. Uh, Again, this was a topic that it will be covered in genetics in more details, or you already covered, and I just want to remind you that viruses are small infectious particles that are enclosed into a protein coat, and they have different nucleic acid. It could have DNA or RNA, and we already know that there are some cases that you can have DNA, or if there is RNA, they are actually able many times to change faster, to make more mistakes, but at the same time, as they made more mistakes, they can vary faster. They are some with a single strain, there are ones with double strains of DNA, and there is a lot of variation, and you have viruses of different sizes, and it's a field that is super interesting, and still we don't know how to define if a virus is alive or not, because it needs a host always. It cannot live without a host, and there is a controversy about how they appear. It seems that as usually there are different theories, but many of them think that viruses appear later in evolution because they require a host to survive. Uh, and there is so much difference in the type of virus, in the sizes of the virus. Here, generally, what you have is you need always an envelope, yeah, a membrane that surrounded the viruses, that you can have different proteins, yeah, that allow them to attach to the cells, and inside generally is the the genetic information, yeah. Uh, some of them have different variation in the types of membranes. So, for example, these are very common that we use a lot. That we talk in genetic revolution. These are fakes. That this one in particular, these viruses affect bacteria. So this is why they are called bacteriophages. And it's a very interesting field that if you want to know more about it, I can send you. I felt tempted to show more information about viruses and iBiology lecture about it, but I contain myself, and I think there is a lot of material. But if you're interested, let me know. Okay, so why we're studying this? Because this is the introduction of a very important topic that is called oncogenes. Yeah. So this the influence of viruses into or the relationship between virus and cancers. It just led to a very important finding that, well, in the beginning I was talking about chromosome rearrangement. Now we're going to talk about particular genes that have a clear connection with cancer. Okay, so I'm going to talk about oncogenes, and later I will. But before that, before talking about oncogenes, I want to tell you about the different stages of cancer and their relation with cell